coffee here. It's a real pleasure to introduce Juraj Bergman as our speaker today. We don't get a lot of talks like this that really are looking at things like in macrogenomics and evolutionary biology and a lot of the amazing technical advances that have been made. I just have to go back when I was a postgrad student, when people were just starting back then to use gel electrophoresis, which must be considered something like a Neanderthal of genetic techniques. But it really is remarkable how far things have come and what kind of interesting, intriguing, and important inferences can be drawn about populations today simply by sampling them and then understanding what's happening, using that to understand what's happening in the past. Juraj is a assistant professor at Aarhus University in Denmark, where it's now 7 o'clock in the morning. So we really appreciate him waking up early to get together with us. His work is very integrative. He combines things like genomic research, macroecology, life history, environmental factors, a lot of other things into a big package to make inferences about mammals and about their evolutionary history in the past. So without further delay, we'll welcome Juraj for joining us here today. Thank you.
Okay, thank you. Thank you for the nice introduction and the opportunity to present my research. I hope you can all hear me okay. So, as Bill mentioned, I am a population geneticist, so I'm not a trained ecologist. I'm not even less a tropical ecologist, but nonetheless, I hope what I present to you today will be somewhat useful and interesting. Okay, so I have segmented my talks into 4 sections. So first I'm going to give a bit of an introduction, and then I'm going to present hopefully 2 of the topics, 2 projects, and lastly I'm going to conclude. Okay, so let's start. So I'm sure you're all aware that Earth has gone through some dramatic events in its past, and it doesn't get much more dramatic than a mass extinction. In the deep past, there have been 5 of these events when the extinction rate went up compared to some background extinction rate of species, and there is pretty substantial evidence that we are either on the brink of or very much in the middle of a 6th mass extinction, which is obviously due to human impact. And this causes a lot of problems, obviously, especially for people who are directly dependent on biodiversity or ecosystems for providing their livelihoods. So this reduction of biodiversity also reduces this ecosystem services, so access to nutrients, water, medicine, etc., for a lot of the people around the globe. So the main question that a lot of us are asking is, how can we address this biodiversity crisis? And there are, many ways to skin a cat, which is probably not the best metaphor to use when talking about biodiversity conservation. Nonetheless, I'm going to try and make a case and show you some results to hopefully demonstrate that we can use population genetics, DNA sequencing to learn about biodiversity and to come up with some baselines or some sort of parameters that are useful for conserving it for the future. So population genetics very much started with these 2 men, which I'm sure you're all familiar with. So we have Charles Darwin and his theory of evolution, where he observed continuous variation in biodiversity in his travels around the globe. He did not, however, know how this biodiversity is inherited from generation to generation. He had some ideas, but not, a very definite structured model. Whereas Gregor Mendel, on the other hand, studied heredity, and he studied it using mostly discrete traits. And he came up with his laws of heredity. So in the beginning, these two theories, so Darwin's kind of gradual evolution and Mendelian kind of discrete inheritance laws, were in opposition. And at the beginning of the 20th century, mostly by work of these three men, these two theories of heredity and evolution got synthesized into what is known as the synthesis of the theory of evolution, right? And these Mendelian inheritance laws. And these three researchers, Fisher, Wright, and Haldane, mostly worked on theoretical models, so they were very kind of mathematically gifted. And to work on these problems, of course, back then in the 20s, 30s, 40s of the 20th century, you didn't have much data to go on. So all you can do is theory, more or less. And it is through the work of, these men that we kind of came up with all these evolutionary processes, or the theoretical background of evolutionary processes. And the methodology that I use today that I'm going to present is very much based on the theory that these guys started. However, back during that time, not much was known about what was the actual unit of inheritance. And this knowledge came about more or less in the 50s, when we discovered the structure of the DNA double helix. And ever since then, we've been trying to sequence DNA to crack this 4-letter code to learn more about organisms. And at first, sequencing was slow going and expensive. So 30 years ago, the Human Genome Project started, and it cost hundreds of millions of dollars. It was a project that stretched over decades, and it was a collaborative effort of dozens of scientific institutions. But sequencing technology has advanced, leaps and bounds since then, and now we are able to sequence a single genome very fast and at a very low cost. So under $1,000, you can get a sequenced genome. And this, again, led to kind of a new expansion of DNA sequencing projects. So we are now moving away from a human-centric genomics projects to more, any type of organism that has any type of organism that can be sequenced. And in the last couple of years, there has been this kind of development of various consortia that have various interest groups corresponding to different organisms, and all of these consortia are under this Earth Biogenome Project. And the aim of this Earth Biogenome Project is to sequence everything, basically, to sequence all known eukaryotic species in a 10-year time frame. And this is, a big effort and a lot of data that will soon become available. And it already is available for us to study population genetics with. Okay, so we're moving towards what has been defined as macrogenomics, where we are looking at patterns, nucleotide variations of genome sequences in different species, and trying to learn something more about, not just species specific, but something that may be a bit more broader, something on the scale of macroecology, for example. And this is very much where my work tries to kind of be. So I'm trying to use large-scale genomics to inform kind of large-scale environmental processes and macroecology of species. Okay, and with this, I'm going to present a first project that I worked on. So I'm going to talk a bit about mammalian macrogenomics. And perhaps the group or subgroup of mammals that have been most affected by extinctions and population declines are megafauna mammals or large mammals. There's been a lot of work on these species. They are very charismatic, very interesting to people. And we also have a lot of good genomic sequences. And there is, let's call it a fierce urgency of now to kind of conserve these species because they are not doing well globally. Okay, but this is not a recent trend. In fact, large animals in general have been going extinct for a while now, and more recent extinctions have also been more size biased. So large species have been preferentially going extinct in more recent periods, especially during the late Quaternary. However, we also have some megafauna survivors that survive to these days or to present time. And the question that I want to ask is, how did these species that survive to present time, these megafauna species, how did their population dynamics look like during this late Quaternary, very turbulent period of megafauna history? And there are two scenarios that can be envisioned. Firstly, these species that survive to present time could have been like, this is great. All of my competitors, large species competitors, are going extinct. I can take up their leftover evolutionary niche, and I can expand in population size. That's scenario one. And scenario two is, maybe these species were also affected by the same forces that their extinct counterparts were. So their population dynamics decreased, or their population sizes decreased during the late Quaternary. In my group, well, I say in my group, it's only a one-person group at the moment because I started it quite recently. Nonetheless, in my group, we have collated a data set of whole genome nucleotide diversity for 550 mammal species. Here you can see a distribution of average nucleotide diversity of these species. They are separated in mammalian orders, and 139 of these mammalian species can be classified as megafauna. So now I'm going to present some results only to do with these 139 megafauna species. Okay, so what can you do with this whole genome diversity maps? So the methodology or the results that I'm going to present are based on this specific methodology called the pairwise sequential Markovian coalescent. And as I said, this methodology is the result of all of that theory that was done 100 years ago, but by very, very clever people. But in kind of interest of time, I'm just gonna tell you what this method does, not how exactly it does what it does. So this method can take the distribution of nucleotide diversity genome-wide. So some sites in the genome will be homozygous. In other words, they will not have a mutation. Some sites will have a mutation. And then this distribution of mutated versus unmutated sites can be used to can be translated into a population size trajectory of a species through time. And you can, maybe conceptually come to terms with this if you think about, when mutations occur. So a lot of the mutations that have occurred that are segregating now in populations have occurred hundreds and millions of years ago. And we can use coalescent theory to classify mutations with respect to time bins when they occurred. And then we can also infer something about specific time bins. And in this case, we are inferring what is the most probable population size that produced this distribution of mutations within this specific time bins. To make a long story short, what you end up with is this plot here on the right. So here you have on the x-axis time, and on the y-axis you have the effective what is known as the effective population size. And each of these lines corresponds to a population trajectory of a single human population. And you can see that there are some variations, obviously, oscillations in population sizes of humans, and there's, as expected, a massive increase in population size of humans in recent times, as we kind of know also from archaeological, paleontological data that humans have massively expanded across the globe. And this is reflected in the genomics of humans. Okay, so now I can return to my question. So I have the data, I have the method. So how did population sizes of this extant megafauna for which we have genomic data, how did they change during the time of this late Quaternary extinction period? And unfortunately, the news is not good. So here on this plot, from left to right, we're going from past to present time. And you can see that there are two periods in megafauna population dynamics during this time. There's this more ancient period where megafauna population sizes were more stable. And then there is a breakpoint here, this blue rectangle. During this time, the megafauna population dynamics start decreasing. Population sizes start decreasing globally across megafauna populations. And so I just want to say that each line is a single species. So there are 139 of these little stepwise lines, and each line is colored by the mass of the species. So what I observe is that with this genomic analysis, that over 90% of extant species, megafauna species, have declined. And the onset of this decline is somewhere between 32 and 76,000 years ago globally. So there is kind of a relatively narrow window in which this decline started. Right. And there is, the next thing I did is I related the slope of the decline, in other words, the severity of the decline with the mass of the species. So the slope in this case tells you, the more negative the slope, the more severely or the more dramatically a reduction of population size the species have experienced. And you can obviously here see a negative correlation between mass and slope, meaning that larger species, larger extant species are the ones that declined more in recent times compared to smaller megafauna species. And this is a significant result because it kind of answers the question we posed in the beginning. So in other words, population dynamics of extant species mirrored the extinction dynamics of megafauna species in general. So size selectivity or size bias is not only observed in extinctions, but it is also observed in recent population dynamics of megafauna species. Okay, and the next question that poses itself is what is the cause of this reduction? There are usually two main causes that are considered. Of course, the first one would be that climate is the main cause of population dynamics in megafauna. And to test this hypothesis, I separated my dataset into two segments. So I just want to say that this is the same plot that I showed you here. These are the same lines, only here time is logged on a log scale, and here this is the natural scale. So here you can see a bit better that the stability of megafauna populations and then a rapid decline in more recent times. And to test whether climate caused this decline, what I did is I separated my data set into a training test set where I used climate-based predictors to model population sizes of this older period. And then I used the inferred coefficients to try and predict what the population sizes would be in more recent time given this model, and then compare it to what we actually observe. And I use different climatic parameters as predictors, so temperature and precipitation, as well as climate lag, and I also assumed different relationship between population sizes and climate parameters. So, for example, a linear relationship between climate or temperature and population size, or maybe a more realistic kind of quadratic relationship where a species achieves kind of, there's an optimum temperature at which a species achieves maximum population size. And so here's the result of this exercise. So here in this upper plot, you can see the observed trend in red. So this is what we infer with genomics, and then this is the predicted trend, which is predicted by a model that is based on this training set of points. And you can see that in more recent times, around between 75 and 100,000 years ago, the model predicts population dynamics quite well. But as we move from past to present time, the model fit becomes worse and worse. Sorry, I was just checking the chat. I was thinking that maybe someone doesn't hear what I'm saying. Okay. In any case, the model that is based on climate is unable to predict population dynamics closer to present time. And this is also reflected in the mean squared error of the model. Okay. Next, what I try to do is to include some human-based parameters. And here now I'm gonna model the whole trajectory. So I'm no longer separating my data set into this training and test set. I'm just interested in the explanatory power of the variables themselves along the whole of this trajectory. And to include human impact as a predictor, I'm using the known or the archaeologically inferred human arrival dates to different land masses. And then I'm also assuming that after humans showed up in a specific area where you have specific megafauna species, then the megafauna trajectory took a turn. It could have started to linear increase or decrease, so decrease or increase. So I'm leaving this open to the model to decide whether or not there is a decrease or increase after human arrival. There are also different types of dynamics I modeled, so the decrease could have been, the dynamics could have been linear or logistic or exponential, etc. Okay, and I have also constructed all kind of combinations of models. So both models that only take climate as the predictors, models that only take humans as the predictors, and then a combination of climate and human parameters. And here you can see in the left the result of this modeling exercise. So here I'm plotting the mean squared error of the model for different time periods. You can see that the climate-only model here in red again has the highest mean squared error, and this mean squared error increases with recent times, whereas models here in blue and orange that include humans as predictors fit the data a lot better than climate-only models. Obviously, the model that combines humans and climate has the lowest mean squared error, but when I punish this model with respect to the number of parameters it includes, it turns out that actually human-only models are the ones that have this highest log score. In other words, these are the ones that kind of least overfit the data and explain the data with the least amount of parameters. So human-only models are the ones that perform best. And here is just a bit of a more visual representation of what the model predicts. So I've taken the best fit for the climate-only model, human-only model, and combined model. So here in the upper plots, this yellow line This is the observed trend from the data. And then the thicker lines in red, blue, and orange, these are the median trends predicted by different types of models. And these gray lines are species-specific trends. Sorry, the yellow line is also a median observed trend, sorry. And you can see that climate model especially struggles in this period, which is around 120,000 years ago. And this is the last interglacial. And if you look at the data, this is where a lot of these megafauna species experience highest population sizes. Whereas during the current interglacial, a lot of, or the majority of megafauna experienced their lowest population sizes. And this is what the model is kind of struggling to fit, this juxtaposition between similarity of temperature between the interglacials and the dissimilarity between population sizes in these two periods. Okay, so I'm going to conclude this part of the talk. Hopefully I've shown you that at least when you're using genomic data, we can see that very likely humans are the main drivers of megafauna dynamics. And we can also use kind of genomics and these predictions of effective population sizes to come up with some concrete estimates of what we have lost. And using this genomic-based predictions, so what I was able to kind of come up with is that we have lost over 1 billion megafauna individuals from global ecosystems over the last 50,000 years. And this can be thought of as an important baseline for restoration. And the loss of this 1 billion megafauna individuals means that we've lost 90% of census-sized biomass and metabolic rate that these species contributed into ecosystems. Okay, and there's a paper. Okay, so I mentioned that I don't have just megafauna species, I have these other mammalian species, and I've also applied this methodology to them as well. Unfortunately, also for non-megafauna species, the trends don't look so hot either. So there's a general decline of global mammal communities, it seems, at least using genomic data. And there's also this dependence on mass that is kind of maintained. So again, larger species are the ones that have declined the most in recent times. Okay, so now I'm gonna talk relatively briefly about another project that is a bit related. So I have shown you that traits affect population dynamics. So far I've only shown you how mass affects the severity of population decline. But of course, there's a bunch of other population dynamics and traits that can be looked at to kind of see how species evolve generally. And generally, the evolutionary success of a species can be measured by aspects of its population dynamics. And classically, population dynamics have been equated to population density, which is the number of individuals per some unit of area. And there is this classical paper by John Damuth where he showed that when you correlate population density with mass, you get a very consistent negative correlation. But there are some kind of caveats to this analysis. And the caveats have to do with using population density. And this population density is usually estimated from contemporary populations, and there are varied methodologies that people use to estimate this parameter. And also it is just a single aspect of population dynamics. We can also look at this effective population size that I've been estimating to see how this aspect of population dynamics is affected by traits. Okay, and this is what I set out to do. And I used a bunch of traits from different mammalian trait databases. These traits broadly correspond into three categories. Some are these classic allometric traits that have to do with species mass. Some are dietary traits, and then I have a bunch of these different reproductive traits. And I wanted to relate these traits to population dynamics of mammals. And of course, the problem that arises naturally from this is that as soon as you have a bunch of these traits, they all correlate with each other, and it's hard to see which trait is actually the one that affects the response variable of interest. Luckily, Sewell Wright, so one of these three guys who was doing theory in the 20s, also had a similar problem 100 years ago, and he basically invented this method of path coefficients, where you can put these traits into specific graphs, so you can relate these traits causally with each other to kind of account for this correlation between traits, and then have a more unbiased estimate of the actual effect of traits on the response variable of interest. There has been developments in this path analysis as well, so it's still very much based on Wright's ideas, but now we have more modern path analysis methods that also take into account phylogenetic relatedness between species. And this is the specific method I'm using here, this phylosim method. Okay. And to do this path analysis, I have to define some trait networks, so how these traits relate to each other. I'm not going to go into too much detail as to why I've pointed these arrows the way I did, because I guess I'm running a bit out of time. But nonetheless, I came up with a trait network, and this is what kind of full model would look like. So you have all these different traits relating to each other, and they're all pointing to this y variable of interest. And I am considering two variables here. I'm taking either population density, this classical measure of population dynamics, as my response, or effective population size, so this genomics-based measure of population dynamics. Okay, and here are some of the general patterns that I have observed. So here in the top plot, you have population density taken as the response variable, and then each cloud of these points corresponds to a distribution of the regression coefficient for one of the 11 different phenotypic traits. So here in yellow, you can see that these allometric traits, so body mass, brain mass, metabolic rate, negatively affect population density. So the larger you are, the smaller population density you will have, which is in line with Damuth's original observation. Whereas if you look at other traits, you don't see that much of an effect. If I take effective population size as a measure of population dynamics, I see different trends. Obviously, there's still a bit of an effect of mass, but there's also the most striking effect is that of sexual maturity. And this kind of makes sense because effective population sizes can be thought of as the number of individuals that actually reproduce successfully to create the next generation. So obviously the lower or the faster you achieve maturity, the more offspring you might be able to produce to contribute to the next generation. Here I am considering all mammals, all different orders together. But of course, different orders of mammals have different life history strategies. So what I did is I tried to separate my dataset into different orders and repeat the analysis to be order specific. And here I'm just going to cherry pick some trends that I observe. For example, an interesting trend has to do with the relationship between population density and brain size. So when I take population density as a response, brain size always correlates negatively with it. However, when effective size is taken as a response, I see quite a different pattern. So especially for carnivores and primates, it might be beneficial to have larger brain sizes to achieve higher effective population sizes. So if you have a larger brain size, you are likely to be more reproductively successful as a carnivore or a primate, even though brain tissue is metabolically quite expensive to maintain. Whereas, for example, if you're an ungulate, you don't really care to have a large brain. You're just probably, your large size is the one that determines your evolutionary success as a species for an ungulate. Okay, I don't need to just look at different mammalian orders. I can also separate my species into trophic guilds, and here I also see a interesting pattern when I correlate animal diet to population density or effective size. Basically, what this shows is that if you're a not as selective, if you are not as selective in your diet, then you will generally do better with respect to both population density and effective size. And I can also separate my species with respect to the biome they occupied. And here I also observe some trends, especially when I correlate, for example, effective size with mass or effective size with weaning age. So mass has a very negative effect if you're a cold adaptive species. If you're a large cold adaptive species, you will have a very low effective size, whereas in the tropics, this relationship between mass and effective size is not as pronounced. On the other hand, you want to have long weaning age to achieve large effective size if you are a cold adaptive species, whereas in the tropical system, you might not be interested in having long weaning ages, so you might want to kind of invest into just reproducing as fast as you can to kind of increase the effective size of the population. Okay, so this is the last take-home message I have. So I hope I showed you that, depending on which population dynamic parameter you choose, you might see different relationships between traits and population dynamics. And this might also be leveraged to improve species conservation strategies. Maybe I don't have time to explain this, but doesn't matter. So here is my conclusion. I don't have anything smart to conclude in the end, just hopefully that I've shown you that genomics is useful in informing macroecology and conservation research. And in the future, I also want to kind of shift my focus to non-mammalian species. Apparently there are other species out there as well. And also there are a lot of more detailed data sets that are more ecologically oriented, such as temporally and geographically stratified climate data that I might want to use in the future to kind of inform these patterns that I've been showing. Okay. And with this, again, I'd like to thank you all for your attention. I'd like to thank all of my collaborators on these projects, and I'm happy to take any questions. Thank you.
Questions? Juraj, I did have a question. Well, a comment really. I was very impressed that your analysis was able to tease apart and show really what appeared to be quite clearly that what looks very much like the human blitzkrieg hypothesis is very much supported. And the climate change hypothesis, which is very trendy these days, of course, and in my personal opinion, it may be over-applied in some sense, people trying to make it fit, but I don't, I actually very much agree with your interpretation. Humans are unbelievably lethal, and we take out the biggest things, don't we? We wipe out the biggest things, and the other things that survive, if they're big, they're in trouble too. So that's
yes.
You'll have to send us a copy of that paper, because there's going to be people who would like to see that, I think. Seriously, yeah, for sure.
Sure.
Any other questions?
Fair enough.
Yeah, Bill, Penny here online.
Go ahead, Penny.
Yeah, I've got some questions. Thanks for the talk. It was really fascinating. I was wondering, you mentioned you were going to use data sets in the future that might account for geography and other things, but I wondered whether your current data sets actually account for a number of human-related things like the late arrival of humans, for instance, in the Amazon and Africa, where humans and megafauna evolved together, and whether or not you took account of the strength of the glacials. Thanks.
Yes, thank you for the question. So with respect to detailed kind of arrival times to specific regions of large land masses, I did not take this into account. So I took, you know, a general estimate around 18,000 years the humans arrived to South America, and I kind of assumed that this is, you know, when they started having their impact. More kind of localized, geographically stratified impact I haven't really considered. And with respect to glacials, so I mean, the temperature parameter that I used and the precipitation parameter I used is a global parameter. And of course, it might be, or it would be definitely better to use something that is more geographically stratified, because glaciation didn't have the same effect in all geographic regions, obviously. So this is something that I kind of definitely need and want to incorporate, so more geographic stratification in general. But for this study, I haven't done this detailed analysis.
And Africa, how do you account for?
Yes, Africa. So Africa, I guess, is special and a bit tricky. So this range that I used, this 130,000 to 200,000 years ago, this is the range that is kind of considered the time frame during which humans expanded throughout the whole of Africa. So this is, you know, a very general, I guess, and the largest time range. So I'm not. So all I'm saying is that, okay, this is when humans have massively expanded and therefore started having the largest effect. Of course, there's all this theory that humans and megafauna have been co-evolving, especially in Africa, and this is why a lot of the megafauna that we still have are African megafauna. But nonetheless, even when I kind of take this quite broadly defined time frame, I still see an effect of humans rather than climate in Africa, in declines of African megafauna. So there is still a human effect there.
Hi, yeah, really great talk. Really enjoyed it. I'm just wondering, did you include any marine mammals in your analyses? And if you did, were there any noticeable differences between the terrestrial and marine mammals?
Not in this first part that I showed. So within the full data set of 550 mammals, I do have marine mammals, obviously. So here, if you look at the end of this plot, so these are all the whales that are above 10,000 kilos in weight. So I do have them in my data set. And when I look at population sizes of marine versus terrestrial mammals in general, there is definitely a steeper decline in land mammals, as you know, you would expect, both with respect to climate and humans. So humans, you know, obviously we didn't migrate underwater, but also climate was probably, or climate changes are probably milder in the marine environment, because, you know, organisms can, I'm just assuming here, can more easily find their kind of preferred temperature in this type of medium compared to, you know, land mammals, I would say.
Thanks. Yeah, it makes sense.
Anybody else? Questions? Nope. Well, Juraj, there was a huge amount of interesting stuff in that talk. It's one of those talks that could be sort of, you could turn it into 6 talks, I think, and you just have, there's just so much there, so much interesting stuff. So yes, if you could please send us a set of your papers, I really would be interesting. And everyone, please join me, folks online and folks here, and thanking Juraj very much for this presentation. Thanks a lot.
Yes, thank you for the invite.